9 o'clock. Shall we start? Okay, I think now we need to start recording. Is that correct here, Ashley?
Yep, we are recording. You can go ahead.
Okay. Hi, everyone. I am Jean Harris. I'm the editor-in-chief of MPMI, and I'm really excited you're joining us here today. While people are still joining, logging on, I just wanted to remind everyone of our next focus issue. This was the top question that came out, the top 10 question of our unanswered questions in MPMI. How do plants engage with beneficial microorganisms while at the same time restricting pathogens? A big puzzle, something a lot of us are wondering about. I hope you will come take a look and think about contributing your work, either a paper or a review for this. I'm now going to stop sharing and we'll move on to our featured thing here. So, okay, well, we still have some people joining here. So as I said, I'm the editor-in-chief of MPMI. We're starting our new series, What's New in MPMI, where we're featuring different authors from who've published recently in MPMI who are going to be talking about their work. So this is another way where you can actually hear it from the author's mouth and ask all the questions that you wanted to ask. And if you want to take a deeper look, all of the papers that are being featured in this series are made open access so that everyone should be able to read those. I'd like to introduce everybody else up here. Mary Beth Mudgett is my co-host. Mary Beth, do you want to introduce yourself?
Hi, I'm Mary Beth Mudgett. I'm a professor at Stanford and I'm the president of IS-MPMI. It's so great to see so many people from around the world logging in. So I think you'll be up for a very nice treat.
And in just a minute, I will introduce our speaker, Jennifer Lewis, who was the first author of our editor's pick in, I believe it was March. A very interesting paper, and you'll see why in a moment. But before we move on to that, I just wanted to say that one goal of this series is to draw people together. We felt that we needed to strengthen connections in our community. Our community spread across the world. Right now, many of us are trapped at home. I'm in my basement here. And so it's nice to be able to reach out and connect with the community. We're not going to meetings, we're not going to conferences. And this is a way for us to find out about what's new, about new research, to meet new people, and to really just build our connections. A big goal of this is to foster inclusion. And so you don't need to be a subscriber. You don't need to be a member of IS-MPMI. I hope you'll share this widely. For those who are not able to join us synchronously, we're going to be recording this and the recording will be available later. I think this is also especially going to be helpful for people for whom English is not a first language. You get a chance to go back, re-listen to parts that were confusing. And of course, since we have just made all these papers open access, you'll be able to go for a deeper dive into the data. So I'm going to move on. Our today's speaker is Jennifer Lewis from UC Berkeley. And her paper caught my eye because it's addressing a really big problem, citrus greening, Huanglongbing, which is devastating citrus crops worldwide. I'm sure you know the numbers better, Jen, than I do. But let's just say, if you want your orange juice, we need to figure this problem out. And big important, not just for consumers, but for farmers. And one of the big puzzles here is how do you figure out what the mechanisms, what the molecular mechanisms of pathogenesis are when you cannot culture the bacteria? And so Liberibacter is unculturable, although as Jen points out to me, it grows just fine in citrus trees. So there must be some way to culture it. And so it's very hard to study. So how do you do this? And she had a very interesting comparative genomics approach that I think really leverages a lot of our knowledge in genomics and bioinformatics to try to address this problem. So anyway, I'm going to introduce Jen. If you'd like to, I don't know, maybe say a word or two to introduce yourself, and then we're looking forward to hearing your story.
Okay, thanks Jean, Mary Beth for the invitation. It's really nice to be able to join you today to talk about our research. I'm a principal investigator at the Plant Gene Expression Center and an adjunct associate professor at the University of California, Berkeley. My background is in viral pathogens as well as bacterial pathogens. And so today I'm really happy to have the chance to talk to you about a project in our lab involving citrus screening, which, as Jean said, is a huge problem. So I'm just going to start sharing the screen now. Okay. So thanks everybody for joining us. It's really nice to be able to have this opportunity to talk about science despite all of the things that are happening in the world, that we can come together and talk about really interesting scientific questions. Today, I'm going to talk to you about our use of comparative genomics to identify peptides that elicit immune responses, and these peptides come from Candidatus Liberibacters species, which are the causal agents of citrus greening. So Huanglongbing, or citrus greening, has really devastated the citrus industry. It's been a huge problem in Florida, but it's also moving into other states in the US. One of the symptoms that you can see is asymmetrical leaf discoloration. And so that means, as opposed to a nutrient deficiency where you would have even discoloration of the leaf, you actually see different patterns of leaf discoloration across the midvein of the leaf. Really critically, the fruit is heavily impacted. So here on the right side, you can see that the oranges are normally very large and they're orange, obviously, and when the fruits are come from infected trees, they're much smaller. And you can see they have this discoloration, so they typically don't color normally, so the bottom part is green. They are often small, they are unevenly round, and really importantly, they don't taste good. They have a bitter flavor. And that's because the sugar content and amino acid content of the juice is affected as well. As well, you have yellow shoots and twig dieback, and ultimately this is fatal. So this is, of course, a huge problem because we're talking about trees here that take some time to reach maturity and start to produce fruit. And by the time you see the disease, you're seeing huge effects on yield. And you can see a graph of that, of citrus fruit production in Florida. And so here on the y-axis, we're looking at thousands of boxes of fruit. And on the x-axis, we're looking at time. And in orange is the total citrus production. And you can see that over a 15-year period, there was a 60% reduction in the amount of fruit. And this impacted both orange as well as grapefruit. And so this has really had a substantial effect. You can particularly see this in Florida, where there's been so little fruit produced that a number of packing plants have closed. And so obviously that has a huge impact for those communities as well. So the disease is quite complex. It's caused by a bacteria called Candidatus Liberibacter asiaticus, or CLAS. But it also can be caused by two other strains, Liberibacter americanus and Candidatus Liberibacter africanus. And they're given the Candidatus status because they are unculturable, at least as of now. They have not yet been cultured. And so scientists are certain that these are quite certain that these are distinct strains, but they have not yet been able to conduct normal microbiology with them. So these bacteria cause disease in a number of citrus species. So here I've given some examples: orange, grapefruit, tangerine, etc. And part of the reason that the disease is so devastating is because it's vectored by an insect, the Asian citrus psyllid, Diaphorina citri. And so the insect carries this bacteria, and that enables the bacteria to spread even more easily through orange groves. And as makes sense with an insect vectored pathogen, this bacteria is also phloem limited. So it's restricted to the phloem, which is itself quite a difficult structure to get to in the plant. And so these combinations of an unculturable pathogen that's phloem restricted on a woody species really make this pathosystem quite complex. So for the purposes of this talk, I just want to focus on one layer of immunity, and that is called pattern-triggered immunity. And this is basal immunity that typically protects most plants from infection. And this is why when we look around at most plants in the world, we don't see them typically infected and diseased. And so this concept will be familiar to those of us in the field, but for the benefit of people who are not familiar with this, and also to have a background for my talk, I just want to give you a little bit more detail about this. So typically we would have conserved molecules from pathogens. These are called MAMPs or PAMPS, and this stands for pathogen or microbe associated molecular patterns that are recognized by transmembrane receptors in the host. And these transmembrane receptors are also called pattern recognition receptors, or PRRs, and they have an extracellular sensing domain for recognition of the PAMP and an intracellular signaling domain to allow the induction of downstream signaling to induce immunity. And these responses lead to pattern-triggered immunity, or PTI. And PTI results in a whole suite of different responses in the host. And some of those examples include the production of reactive oxygen species, or ROSs, PTI marker gene expression, so the expression of genes that are associated with immunity, callus deposition, and ultimately enhanced resistance to the pathogen. And so we thought that this concept of small molecules, peptides that derived from the pathogen that could result in immunity could be a really interesting way to tackle this problem of citrus greening, because it does not require culturing the pathogen. So we decided to try to see if we could identify putative PAMPs or MAMPs from Candidatus Liberibacters species that would elicit PAM-triggered immunity in citrus. So to do that, we decided to take a comparative genomics approach to identify these PAMPs. So I want to also remind you that PAMPs are typically derived from highly conserved proteins. And since they're highly conserved, we infer that they are essential for the pathogen. And so Claire Bendix, as a postdoc in my lab, looked at the genomes of different Liberibacter strains that were available at the time. And this included 3 different Liberibacter strains that infected citrus, the ones I mentioned at the beginning, Asiaticus, Americanus, and Africanus. A strain, Candidatus Liberibacter solanacerium, that infects potatoes. And then a non-pathogenic relative, Liberibacter crescens. And so she looked at the genes in these genomes to extract the core genome of these strains. And then we looked to see if we could identify different patterns of selection. So the premise for this was based on the work of Honor McCann a number of years ago. And so they proposed that if you had recognition by the host, you would have strong selective pressure to diversify parts of the PAMPs that were not essential for the activity of the PAMP. As I already told you, typically these are essential. But if you evolve the parts that are not essential, you could see that as positive selection. And so here we looked basically for parts of the proteins that were in a background of negative selection, but had individual residues that were under positive selection. So this gave us different criteria for trying to prioritize which PAMPs we would actually test, which peptides, I should say, we should test. And so the kind of general criteria we used is that they should be under negative selection and have signatures of positive selection. They should be specific to citrus-induced infecting strains. They should be potentially absent from strains that infected the Solanaceae, and they should be absent from non-pathogenic strains. And so using these criteria, we ended up with a list of 13 different genes that carried peptide regions that had these patterns of positive selection. And we particularly focused on ones that had a large number of positively selected sites, although we also included flag G as a candidate peptide because it's part of the flagella, and flagellin, as we know, is a very strong inducer of pattern-triggered immunity. So then Dr. Yuan Chen joined my lab as a postdoc, and she did all of the experimental work that I'm going to talk about today. And so she used a peroxidase assay that was developed by Adam Mott to look at the production of reactive oxygen species in well-characterized systems to begin with. So we started off testing in Arabidopsis and Nicotiana benthamiana because we wanted to see if we could actually validate that these would induce reactive oxygen species production. So we treated the leaves of these plants with the candidate peptides, and then we used this colorimetric assay that measures apoplastic peroxidases and produces a substrate that can be measured in a spectrophotometer. And so here I'm showing you the data for y-axis. You can look at the absorbance of the colorimetric substrate. And then on the x-axis, you can see the different peptides we tested, and also Murashige and Skoog basal salts as the negative control for the buffer. So as expected, FLAG22, which is a very strong inducer of pattern-triggered immunity, caused a large production of reactive oxygen species. And interestingly, we saw that two peptides, PKSG and NuOD, were also able to induce high levels of reactive oxygen species. So Yuan noticed that PKSG had a large number of hydrophobic residues, and she thought it was possible that this peptide might not be appropriately solubilized in MS buffer. And so through some research, she found that if she put this in a basic buffered PBS buffer, that the peptide was actually more soluble. And so here you can see that we can drop the peptide level multiple times, and we still get the same level of reactive oxygen species produced compared to FLAG22 in the PBSA buffer. So this work showed that NuOD and PKSG could trigger reactive oxygen species production in both Arabidopsis, and I'm not showing you the data here, but we also showed that we could do that in Nicotiana benthamiana using this peroxidase assay. So the peroxidase assay is extremely sensitive, and it's an endpoint assay. So it looks at the total amount of reactive oxygen species that are produced. But we also wanted to look at the time course of reactive oxygen species. And so for that, we use the luminol assay, which is commonly used to measure the production of reactive oxygen species. And so the basic setup of this is very similar. We would treat the leaves with the peptides, but in this case, we have luminol and horseradish peroxidase producing a luminescence signal that can be measured. So here on the y-axis, you can see the luminescence signal, and on the x-axis is the time course. And this assay is not as sensitive as the peroxidase assay, but it's very commonly used in the literature. And so you can see that PKSG induces high levels of reactive oxygen species production in both Arabidopsis and Nicotiana benthamiana, and that is similar to or higher than that seen with FLAG22 in the basic buffer. We all thought to ensure that the peptide concentration was not sufficient to induce RRS production, we also tested the THRS peptide, which did not elicit any ROS production. And you can see there is almost no signal from that. And similarly, the buffer alone is not able to induce the production of reactive oxygen species. So using these two assays, the peroxidase assay and the luminol assay, we found that PKSG could elicit similar levels of ROS production as FLAG22, and that high peptide concentrations are not sufficient to induce reactive oxygen species. So of course, the big question is what happens in citrus? So we decided to test a wide variety of different citrus species, and these were selected based on reports of either susceptibility or tolerance to HLB. So the species that I've shown here in green, Mexican key lime, Australian finger lime, and Eureka lemon have been reported to be HLB tolerant. The ones in orange, Clementine mandarin, Mineola tangelo, Rio red grapefruit, and Valencia sweet orange are thought to be HLB susceptible. And then the ones in black, kumquat, Kizu mandarin, and mandarin quat, there have been mixed reports. So in some cases they have been tolerant, and in others they have been susceptible. So we decided we would test all of these species for their ability to produce reactive oxygen species, but we needed to first validate that we could actually visualize that in citrus. So to do that, we decided to use FLAG22, which is known to be a very strong inducer of PAMP-triggered immunity in multiple different species. And so we tried using the peroxidase assay, and we were unable to get any production of reactive oxygen species using the peroxidase assay. So we mainly used the luminol assay for these experiments. And then we tested these diverse citrus species that, as I said, have been reported to be either tolerant or susceptible to citrus greening. And so here you can see all the different species that we tested, and in particular, kumquat and Australian finger lime, we saw high levels of reactive oxygen species production in response to FLAG22. And Mineola tangelo, by contrast, we had basically had no responses to FLAG22 that were similar to from the water control. And then the other species had sort of varying levels of reactive oxygen species production in between those two. So this told us that the assay could work and we could actually see reactive oxygen species production in citrus. So then we went on to test PKSG. And here I'm showing you the 3 species that did induce higher levels of reactive oxygen species production. So in Australian finger lime, Eureka lemon, and kumquat, we saw high levels of ROS production that were comparable to that seen with FLAG22. In some cases, they were actually higher than that seen by FLAG22. And again, the basic buffer, we saw no reactive oxygen species production with buffer alone. So then again, this indicated that PKSG could trigger ROS production in these different species, and it was similar to the response by FLAG22. So we can also visualize ROS production at the place that the hydrogen peroxides are produced. And to do this, again, you would treat the leaves with peptides and then use nitro-blue tetrazolium staining to visualize the ROS production. And so here you can see the NBT stained leaves of Australian finger lime. And for FLAG22 in the basic buffer or PKSG in the basic buffer, you can see the quantitation on the right here that we have similar levels of staining in those two with those two treatments, and these are significantly different from the buffer-only controls, as well as the peptide that was unable to induce ROS production. We also did a similar analysis in Mineola tangelo, which, as I already said, does not induce ROS production in response to these peptides. And you can see that these leaves do not respond at all to any of the peptides or the buffer, which is consistent with what we had seen in both the luminol assay in citrus. And so from these data, it was quite clear that FLAG22 and PKSG were inducing similar amounts of ROS in Australian finger lime, and that there was no ROS production in Mineola tangelo. So far I've told you about ROS production. We wanted to look at other ways that PTI can be induced. And so to do that, we looked at different marker genes that are associated with pattern-triggered immunity in plants. And we did this using quantitative RT-PCR. And we selected 3 different marker genes in citrus. These are marker genes that have previously been shown to be responsive to different pathogens, primarily Xanthomonas pathogens in the citrus. And so on the top here, we're looking at Australian finger lime and the relative expression. And then on the x-axis, we're looking at the different buffers as well as the peptide treatments. And so PR1, when treated with FLAG22 in either buffer, there was less expression of PR1 in Australian finger lime, which is consistent with what has been observed previously in different citrus species that were tolerant to canker and HLB. And PKSG did not have any effect on PR1 expression. So we then turn to WRKY22 expression, and here we actually see that WRKY22 is induced in both FLAG22 application as well as PKSG application. And then the last PTI marker gene, GST1, had no effect. Its expression was not affected by the treatment of either FLAG22 or PKSG. And in all cases, you can see that treatment with a negative control peptide THRS had no effect on expression. And so to validate that these data were again due to something unique in Australian finger lime that was inducing resistance, we looked again at Mineola tangelo. So I'll remind you, this is the species that did not elicit ROS production to PKSG. And you can see that for all 3 of these marker genes, PR1, WRKY22, and GST1, we saw no differences in expression upon treatment with any of the peptides. So this is consistent with what we saw with the ROS production, that it looks like PTI marker genes are actually induced by PKSG, although the pathways seem to be different between FLAG22 and PKSG. So of course, then the big question is what happens to bacterial replication when you treat with a peptide? And ideally, this should be done in citrus trees that are infected with Liberibacter. But this disease takes many years to actually develop. And so those experiments are ongoing, but for the purposes of actually being able to do this in a more timely fashion, we decided to treat Arabidopsis plants with Pseudomonas syringae, which is a gram-negative bacterial pathogen that's unrelated to Liberibacter asiaticus. And so here we pre-treat the plants with either FLAG22 or PKSG, and then we carry out a bacterial growth assay using Pseudomonas syringae and Arabidopsis. And so the bacteria and the peptides are both pressure infiltrated, but at different time points. So again, on the y-axis, you can see the log growth of the bacteria, and on the x-axis, the peptides in the different buffers. So as expected, when you pre-infiltrate with buffer and then measure log growth after 2 days, you can see about log 6.5 growth of Pseudomonas syringae and Arabidopsis. When you pre-treat with FLAG22 in either water, the basic buffer, or PKSG in the basic buffer, we saw about a 0.5 log to a 1 log reduction in bacterial growth. And this is again specific to peptides that are inducing pattern-triggered immunity. So when we treated with THRS, we saw no effect on bacterial growth compared to normal conditions. So again, this indicates that FLAG22 or PKSG are both able to induce pattern-triggered immunity and restrict bacterial replication in several different hosts. So for PKSG, we're actually seeing an effect on PTI in Arabidopsis, Nicotiana benthamiana, and citrus, which are all found in very divergent plant families. And so this indicates that PTI is induced in response to PKSG, and it validates that PKSG is a real PAMP. So today I told you about our comparative genomics approach to identify 2 peptides that elicit immune responses. So NuOD elicited ROS production in both Arabidopsis and Nicotiana benthamiana. PKSG elicited ROS responses in both Arabidopsis, Nicotiana benthamiana, but in addition in several citrus species. As well, PKSG elicits PTI marker gene expression in citrus. And when we applied PKSG to Arabidopsis, this resulted in pattern-triggered immunity against Pseudomonas syringae. So we think that this might be a way to help protect citrus plants from infections. So by applying these peptides, we might be able to help boost plant immunity. And this could be another tool to help growers to actually save some of their production from citrus greening. And so we are collaborating with Dr. Bill Dawson at the University of Florida to carry out field testing of peptides in sweet orange. And so Bill has developed a viral delivery system using citrus tristeza virus. And so this system allows us to deliver peptides to the phloem. And this is important because citrus tristeza virus is also a phloem-restricted plant virus. So this would be delivering the peptides to the place where the pathogen is present. And then there's data from other labs to suggest that there is movement of small molecules to the phloem that could potentially induce PTI in other places in the plant. And then this work also brings up bigger questions of how do Candidatus Liberibacters species avoid pattern-triggered immunity. So in many other systems, effectors are important for suppressing pattern-triggered immunity, and this is how they're able to actually cause disease. And in this system, that's also possible. Many labs have done work to try to identify effectors in Liberibacter species. It's also possible that by localizing to the phloem, they're occupying a niche that maybe is not as efficient in triggering pattern-triggered immunity. So I want to acknowledge the people who did the work. Dr. Claire Bendix did all the bioinformatics that I talked about, and Dr. Yuan Chen did all the experimental work that I talked about. It was a really fun project to do that's very different from what the rest of my group does, and we had a lot of great discussions with all of them. And also the benefit of many undergrads. Our collaborators are Bill Dawson and members of his lab at the University of Florida. And funding in my lab is supported by the USDA and NSF. And I also want to mention the PGC greenhouse staff who were really helpful in growing all these different citrus species for us. And while you're busy typing in questions in the chat, I also just want to mention that I've been really fortunate to work with an amazing team of really collaborative scientists. These are just the people I've worked with at UC Berkeley. And they are staff scientists, postdocs, graduate students, rotating graduate students, undergrads, and students from two-year colleges. And they bring a real diversity of gender, ethnicity, race, nationality, experience, LGBT, perspective, opportunity, and culture. And by incorporating these principles of diversity, equity, and inclusion in my lab, we've really sought to foster a culture of scientific excellence. So with that, thank you, and I would be happy to take any questions.
Thank you, Jen, for a very, very interesting talk, and also for reminding us the strength that we bring to our science from having a diverse team to work with. So we are already having some questions. So I don't know whether you want to turn off sharing.
Yeah, I'll do that now.
Okay, yep, people are typing away. Okay, so maybe I'll start the first one, and then Mary Beth, you can jump in. So Tanya Taruno says that the ROS induced by flag 22 in Arabidopsis is quite low. Whoops, sorry, it's moving as people type. Wait, did that one go away? Oh, that's an answer. Sorry, I'm getting used to this question and answer. I hope everybody's typing in question and answer. So The ROS is quite low, and she's wondering whether this could be due to the high concentration of FLAG22 used, 1 micromolar instead of 0.1 micromolar that many labs use. And if so, do you think higher PAMP concentrations might be inhibiting the ROS reaction, and thus you might have missed other eliciting PAMPs?
Interesting. So I think it partially depends on the substrate that people use. And so we just use the normal luminol substrate, but there are some labs who use very sensitive luminol substrates. And in those cases, they may find that lower concentrations of PAMPs are sufficient to induce those responses. We have typically found that 1 micromolar induces a reproducible response for FLAG22. It does also depend on the concentration of the buffer, I should say, that you resuspend the peptides in. And so in our case, because PKSG was very hydrophobic, we had to put that peptide into a different buffer, and the response from FLAG22 is actually reduced when you put that into the buffer as well. So, I mean, it's possible that we missed some peptides. We certainly could go back to those and retest. The POX assay, I should say, though, is also quite a bit more sensitive than the luminol assay, and so that should have theoretically pulled up any PAMPs that were able to be recognized in at least Arabidopsis and Nicotiana benthamiana.
All right. Well, Tanya had a follow-up question, and that was any speculation, given that you think it's a PAMP for these peptides, what the PRRs might be for PKSG or NUD. And then Roger Innes also along those lines was sort of asking the question of, well, if these are in the phloem, how do you perceive these PAMPs getting outside and being detected by these PRRs?
Right. So I think that's definitely an interesting question. It's not something that we have followed up on at this point, but I think that could be done very easily. And I think because you can see responses to both of those peptides in Arabidopsis, you could certainly use the collections of receptors to try to determine which PRR is recognizing either one of those PAMPs. And so, yeah, the question about from Roger about how they would be detected, this is a really interesting question because the phloem is not thought to have a lot of proteins that would be able to recognize PAMPs in this way. However, we know from viruses that they are using the phloem to move systemically through the plant, and then they're unloaded in other tissues. And a lot of small molecules kind of go along for the ride through the vasculature. And so you could imagine that some of these peptides could potentially follow the sink to source transition and actually move into cell types that would have PRRs, and that would elicit immune responses. Now, I think that's a really interesting question. It's not something that we have followed up on, but I think that it really raises the question of how this pathogen was able to avoid immunity, and perhaps the niche that it occupies is one of the reasons for that.
So Sonali Roy has an interesting question looking at the relationship with growth. She asks, does the peptide have any effect on growth of the plants, given that FLAG22 can hamper plant growth? And would that affect yields? What's your thoughts?
Right. So in all of the experiments that we've done, we have had short incubations with peptides. So, so that is effective for the purposes of trying to identify PAMPs and look at the immune responses. You could imagine the constitutive expression of a PAMP could have an effect on yield if immunity is constitutively induced. That has been seen, of course, for other systems where they try to constitutively cause systemic acquired resistance, for instance. So I think that so the way that we have tried to approach this is to see whether expression of the peptides in citrus in the field would have an effect on protecting the plants from Liberibacter pathogens. But it might be necessary to put those under promoters that would be pathogen inducible, for instance, so that they would be a little more restricted in their expression so that you didn't have a yield effect. But at this time, it's too premature to know whether that will happen.
Jen, Kayla Pennerman says, thanks for a great presentation, but also would like, similarly along those lines, clarification about future work. So would you be testing variants of peptides that may be eliciting stronger PTI, inducing more of a wild type threshold to push the immune response? And a follow-up to that, I was just kind of curious, have you looked at variation amongst the strains that you have to see about the mutation rates? Because that was your initial premise, right, that they'd be under negative selection.
Right. So, so we have been testing the peptides in citrus, and we tested all of the ones that I mentioned at the beginning that we've been focusing on. So some of those we could not see a response in either Arabidopsis or Nicotiana benthamiana. So I think so that for at least some of these, they may not have any effect on immunity in citrus. So in some cases, those will be, I would suspect, more of a negative control. And then the ones that we'd be particularly interested in are NuOD and PKSG. And I just want to also mention that disease in citrus from citrus screening takes many years to progress. And so I think we have been working with Bill for the last 3 years to do these experiments, and they are still ongoing. So it's really not as straightforward as, you know, trying to test in a non-woody plant with a fast generation time, and that does limit how quickly you can test different peptides and see the effect. And then, sorry, Mary, oh, you were asking about variation in the peptide. Yeah, so we had started to look at that. The genome sequences that we have, there are not too many of them. I know that Guido and Wembo have now published a much larger collection of Liberibacter strains from many different places. And so that I think would be an even better data set to look at and look at variation. There was some variation for the peptides, but I think, you know, with even more strains available, we would see more of that.
Great, thanks.
So we do have we're trying to use the Q&A feature rather than the chat, but we do have 1 or 2 things that popped up in the chat. So Yasser Nahela asks, how do these peptides interact with plant hormones, particularly those associated with PTI such as SA?
Yeah, so we did not specifically look at plant hormones and look for interactions or effects generally on genes associated with plant hormones. I will say for PR1, we did not see an effect on expression of PR1 in response to PKSG, and that's distinct from what's seen with FLAG22. So that would suggest that the responses that are induced by FLAG22 and PKSG are distinct. So it's possible that it's different, not as really, or at least from, if you only look at PR1, perhaps it's not SA related, but I wouldn't say that we've looked at enough marker genes to really have a firm conclusion about that.
Next question comes from Lace Marina Granado, asking about, what do you think about the phloem clogging? Could that be a PTI response?
Yeah, so there's a lot of speculation about that, that there's more callose deposition in the phloem, and so that may be part of the reason that there's this problem with the photosynthate moving through the phloem to the rest of the plant. It's not really clear if that is a slow immune response and it doesn't happen quickly enough to prevent the pathogen from establishing. The disease typically takes 1 to 2 years to develop, and it can be very hard to tell that the trees are actually infected. You have to use quite sensitive methods to detect that. So I think it's not entirely clear at this point whether there is an immune response and it's just not happening fast enough to stop the pathogen.
So Louis-Philippe Meyer has two questions. I'm going to ask the first for now. Since Liberibacter supposedly primarily colonizes the phloem, and your experiments were done infiltrating into the leaf apoplast, were you able to observe defense responses such as NBT or callose staining when feeding the peptide via the vasculature? So petioles and elicitor dilutions or uptake via transpiration?
Those are great questions. They are not things that we have tried to do, but I think that that would be a logical follow-up to do those things. We did primarily focus on leaves. As you said, we have not tried to focus on uptake through the phloem. So that's unknown at this point.
So Huanglongbing, I don't think we did this one yet, do we, Jean? It was a question about the comparative genomics approach to identify these. Could you use such an approach for fungal pathogens as well, for things that are challenging to get at, you know, their epitopes?
I think you could, if you had enough genome sequences from different strains, and ideally from strains that both cause disease and were non-pathogenic. I think that it could be a strategy to do that. You would have to be able to identify the core genome and then look for those selection patterns. But I mean, part of the reason that we thought of taking this approach is because it does not require culturing the pathogen and doing normal microbiology. And so I think if you had fungal pathogens that were also difficult to manipulate, this could be a good strategy for that.
Claudia Martinez thanks you for the presentation and asks whether you know the activity of the peptides.
The activity in terms of what do they do normally? Is that the question, or?
Maybe.
Yes.
Yes, okay. So PKSG is part of a polyketide synthase complex. And so these are primarily involved in the production of secondary metabolites, and they can also be involved with communication with other bacteria. Other than that, the true function of PKSG is not really known. So we could speculate that perhaps as the populations of bacteria are kind of living and dying, they might be releasing small peptides, and those could be types of things that are recognized by the plant. But we don't know what PKSG would do in the context of Liberibacter asiaticus. It is found in many species of bacteria, and it's also found in archaea. So it's not rare.
So Jen, along those lines, Caitlin Allen is asking about PKS variants. So if you look at the other microbes, do they have PAMP activity in Arabidopsis? Have you tested that either on other plants as well?
We have not tried other variants. This was a side project in my lab. So, you know, this was something that we got interested in, and we thought it would be an interesting question to pursue. But we certainly have not, you know, gone down every possible route that could be explored with it. So I think there's still a lot of open questions, and that's one of them.
Great.
Alejandra Munoz asks, are the genes that you selected totally or partially absent in Liberibacter crescens and solanacearum?
Yeah, so we looked at that and we saw actually relatively few genes that were shared by all of the different strains. And so so if we tried to look at the core genome across everything, there were actually very few of them. And so these are peptides that are specific to the citrus infecting strains, and they're absent in Solanacearum infecting strains and or the Liberibacter solanacerum, as well as Liberibacter crescens.
So there was a similar question from Sheila McCormick or Paul Hertzmark. Did that answer your questions about what were the characteristics of the peptides that were not in the solanaceous pathogens? I think that was a similar, you know, the assumptions you made were, I think, just well described. Jessica Forret has a question about thinking about where experiments in Arabidopsis where you're priming with a peptide and challenging with the bacteria. Is this being done with the same leaves, or are you challenging leaves with bacteria that were untreated with peptide? And in addition to PTI, could these peptides be inducing SAR, systemic acquired resistance?
Right, so we are challenging and doing the infections on the same leaf. So this is the kind of classical assay for PTI that was described in Cyril Zipfel's lab. We are not looking at systemic acquired resistance here. So the time frame for systemic acquired resistance would be much longer than what we're looking at. We're only looking at bacterial populations 2 days after infection, and that would happen 24 hours after application of the peptide. So we really can't say whether systemic acquired resistance is being induced in this case or not. But at least in the time frame that we looked at, it does seem to be a more short-term response.
So I'm now going to read Louis-Philippe Meyer's second question, which is, on a different note, did you try to estimate the EC50 value for these novel peptides? How sensitive is the perception system in citrus species?
Yeah, so we did try, so we didn't completely look at an EC50 value. We did try a range of different concentrations in both Arabidopsis and Nicotiana benthamiana, and those concentrations were quite a bit lower than they are in citrus. Citrus, it is challenging to get the peptides in because of the waxy cuticle. And UN did try increasing the peptide level significantly, and that really had no effect on the level of ROS production. So we don't think that more peptide would necessarily induce a stronger response.
So I will just want to encourage people to write more questions. I have my own questions, so I'm going to ask mine now, but this is a chance for you to ask, right? So you don't always have the chance to have the author right there to ask the question, so this is your opportunity. Type away. While you're typing, I'm going to ask Jen, you keep referring to the fact that this is a side project in your lab, and so I'm curious, what led you to this little side project? How do you get there?
Yeah, so I was interested in evolutionary approaches to try to understand plant pathogen interactions. And I was also familiar with Honor McCann's work, and she used this type of approach to identify PAMPS in Pseudomonas syringae, which obviously is a very well characterized pathogen in our field. And so I was really curious to see if we could take principles of molecular plant pathology and try to apply them to this really significant disease of citrus greening. And it seemed that if it worked, it could be very effective because, as I said, you don't need to worry about culturing the bacteria. You can synthesize the peptides, and so that it might actually have a real impact on the citrus growers. And so that's why we decided to try and see if it would actually work. And we were really excited and pleased to see that it was successful.
Yeah, it's always interesting to see what leads people off on little directions, different questions. No other questions. Do you have any questions, Mary Beth?
I think I'm good. Nope, I think they were all answered.
Yeah, we had some really good questions out there, trying to get at all these different angles. It's such a curious question. Anyway, so why don't we stop and thank Jen again for an excellent talk.
Thank you.
Very interesting. And I hope that you will join us 2 weeks from now. And we're going to be shifting, as you've noticed, between a time that's convenient for the Atlantic and one that is convenient for the Pacific. And so the next one we shift back to the Atlantic, And we're going to be featuring a technical advance, a really interesting story, a collaboration between John McDowell and David Hake, those two labs using long-read sequencing technology to learn something about the genome of Phytophthora. And so again, this is using a genomic approach to really try to uncover some really fundamental aspects of pathogenesis and evolution. Okay, well, thanks everyone. Roger says thanks. Goodbye, and we'll see you in a couple of weeks.
Shout out to
Thank you very
Zoe.
much. Bye.